Hey Bio20s, I'm back again for another lesson. Do you ever get the feeling like someone or something is watching you? Oh, I completely forgot I left these here. I'll go put these cow eyes away. Okay, now we can start our lesson today talking about anaerobic respiration. So this is cell respiration where oxygen is not present. Now, in this case, there's only going to be two processes involved. There's still going to be glycolysis, and there's going to be some type of fermentation afterwards, either an alcohol fermentation or a lactic acid fermentation. But the main key here is that no oxygen is present. Since there's no oxygen present, you can't do the electron transport chain, you can't do Krebs cycle, and you can't do pyruvate oxidation. You, yes, can still do glycolysis, but you're going to have to do something different afterwards. So stage one is going to be glycolysis, the same process we've talked about for quite some time, where we take our glucose, we split it into two pyruvates, and we net 2 ATP. The second stage is then going to be a fermentation. Both of these stages will occur in the cytoplasm of the cell. And once again, there will be no pyruvate oxidation, Krebs cycle, or ETC. Okay, so in alcohol fermentation, this occurs in organisms where they produce alcohol as their byproduct. This would be different types of bacteria. What happens here is we are going to take our pyruvate that we create at the end of glycolysis. We are then going to create a acetaldehyde alcohol. Now the purpose of either fermentation, whether it's alcohol fermentation or whether it's lactic acid fermentation, the purpose is to essentially just keep glycolysis running. That's the whole point. Oxygen isn't present, so the main producer of ATP has to be glycolysis. So what we're trying to do is recycle the NAD+ to keep that glycolysis running. So at the end of the day, when we finish alcohol fermentation, we should end up with 2 molecules of ethanol, 2 molecules of CO2, as well as some ATP. So it kind of looks like this. Glycolysis remains the same. Our 1 molecule of glucose becoming 2 molecules of pyruvate. We get a net of 2 ATP, because remember we put in 2, we gain 4, so a net of 2 ATP, and we get 2 NADHs. Now the purpose of the fermentation step is to take those 2 NADHs and turn them back into NAD+. So what we're going to do is we're going to take our 2 pyruvates, we're going to add 2 molecules of water, and we're going to take out 2 molecules of CO2 to form 2 acetaldehydes. We're further going to take those 2 acetaldehydes and convert them into 2 molecules of ethanol. By doing that, we're taking the electrons and the hydrogen from the NADHs and turning them back into NAD+, which can go back up and essentially keep glycolysis running. So obviously 2 ATP is not a lot of energy. It's not a lot of energy for most molecules. Now some tiny little bacteria, they can live on this and that's fine. We can actually use this to our advantage though. So some processes, like the process of making things like beer, wine, other fermentation processes, sauerkraut, they involve this process. And what you want to do is essentially make sure that everything is sealed properly so the bacteria that you're using has no access to oxygen. You don't want any oxygen getting in there or else your fermentation is not going to work properly. So then we have things like your fermentation products being created, and if there's no oxygen, you're going to have a nice fermented product, whatever that ends up being. Also, this process is used in very simple processes like baking bread. So you put a little bit of baker's yeast right into your bread. One of those byproducts would be alcohol, which essentially gets burned off, and the CO2 that's produced is what allows the bread or the baking to rise. Now, this occurs in small organisms like bacteria. If this happened in big animals like mammals or humans, we would essentially get drunk every time we work out, which is not the case. Would not recommend working out drunk. I don't think that's a great idea. What we do instead is we create a product called lactic acid. If you've ever done a big workout, if you've ever gone to the gym or done something physically active, and then right after, a few days after, your muscles, they feel a little stiff and sore, that is because of lactic acid. Now in an animal muscle cell, yes, oxygen is obviously present. We want to do aerobic respiration. We want to get as much ATP as possible, but sometimes our muscles, they need more. Aerobic might not be enough to get us by. We might have to do a little anaerobic respiration as well. So what we're going to do is we're going to go through glycolysis. We're going to keep that glycolysis running, but by doing so, we're going to produce lactic acid in the process. We're not making ethanol, so just to be clear, we're not making alcohol, and we're also not making any CO2. We are just making lactic acid. So this would occur when you're doing something physically active, right? Doing something physically active, and you're kind of burning through all that ATP very, very quickly. Your muscles are like, hey, I need more, I need more, I need more. You're gonna go through your glycolysis, make your 2 ATP, make your 2 NADH, make your 2 pyruvate, but then you're gonna take that pyruvate, grab electrons and hydrogen from NADH, and create lactic acid. And then reuse that NAD plus to keep glycolysis running. Now, in either case, lactic acid or alcohol, not a very efficient process. 1 molecule of glucose giving you 2 ATP is not very good. It's like if you gave me $100, I gave you $2, very bad conversion. But if that's all you can get at the time, if some ATP is better than no ATP. So when you combo, let's say you're working out, you combo your aerobic respiration with the little extra you get from anaerobic, it may be enough to, do what you need to do in that moment. Now the good news is the lactic acid can go away. It actually can be converted back into pyruvate once your exercise is finished. So yes, it will make your muscles fatigue, get stiff and sore, but if you stop the exercise, then the best thing you can actually do is not just completely stop, do a little bit of a cool down, maybe do a little, short walking cool down, go on the bike, do some stretching, whatever you need to do. That will actually help convert that lactic acid back into pyruvate, and then you can continue on with aerobic respiration afterwards. So that is it for cellular respiration, the 2 different types of cellular respiration. Next lesson, we're going to move on to Unit D, which is human systems, and we're going to begin with our very first system. We're going to talk about the digestive system. So thanks again for watching, and we'll chat soon.